Today, I'm excited to present our latest research titled Transcriptome-wide Association Identifies KLC1 as a Regulator of Mitophagy in Non-syndromic Cleft Lip with or Without Palate. Non-syndromic cleft lip with or without cleft palate, NSCLP, is one of the most common congenital craniofacial anomalies, occurring in approximately 1 in 700 live births worldwide. It is caused by the incomplete fusion of facial processes during development and is influenced by a complex interplay of genetic and environmental factors. Despite numerous studies identifying susceptibility loci through genome-wide association studies, GWAS, the genetic mechanisms underlying NSCLP remain incompletely understood. To gain a deeper insight, our study utilized transcriptome-wide association studies, TWAS, integrating expression quantitative trait loci, eQTL, data with GWAS data to identify key genes and regulatory mechanisms associated with NSCLP. In this study, we used TWAS to identify KLC1 as a key gene involved in the development of NSCLP. The study showed that the SNP-rs1288480G2A enhanced the binding of the transcription factor ELK1 to the KLC1 promoter, leading to increased KLC1 expression. Additionally, we found that the alteration in KLC1 expression subsequently impacts mitophagy, leading to significant changes in cellular processes such as proliferation, migration, and apoptosis, as well as zebrafish morphology. We performed a two-stage GWAS of NSCLP, including 1,069 cases and 1,724 controls, analyzing 3,620,909 common variants. Meta-analysis revealed significant associations with NSCLP at loci 1q32.2, 2p24.2, and 17p13.1. Additionally, we conducted both cross and single-tissue TWAS, identifying 20 genes significantly associated with NSCLP across multiple tissues. Among these, KLC1 was newly identified as a candidate gene. To further investigate the genetic regulation of KLC1, we focused on the SNP-rs1288480G, which was related to NSCLP risk. The colocalization analysis between GWAS and eQTL data revealed a significant association between rs1288480G and KLC1 expression in whole blood. Functional analysis demonstrated that rs1288480G is located within the KLC1 promoter region and affects its activity by altering the binding of the transcription factor ELK1, which in turn regulates KLC1 expression. We next explored the biological role of KLC1 in craniofacial development using zebrafish models with CRISPR-Cas9-based knockdown and overexpression of KLC1A. In the knockdown group, zebrafish embryos exhibited abnormal development, including craniofacial defects, smaller palatoquadrate and ethmoid plate dimensions, and reduced survival rates compared to the control group. Conversely, overexpression of KLC1A led to normal craniofacial development and increased body size. These results suggest that KLC1 is essential for proper craniofacial morphogenesis. At the cellular level, KLC1 knockdown in human oral keratinocyte, HOK, and human palatal mesenchymal, HEPM, cells resulted in decreased proliferation, migration, and increased apoptosis, while KLC1 overexpression had the opposite effect. Moreover, the gene set enrichment analysis using GWAS data revealed only the mitophagy pathway as statistically significant, and we observed that mitophagy hub genes were upregulated in response to KLC1 knockdown. Further, KLC1 deficiency led to increased mitophagy in cells, as evidenced by enhanced colocalization of lysosomes with mitochondria and GFP-LC3 with mitochondria. Transmission electron microscopy and JC1 staining confirmed the upregulation of mitophagy in KLC1 knockdown cells, while overexpression of KLC1 reduced mitophagy. Treatment with the mitophagy inhibitor MDV1 partially rescued the effects of KLC1 knockdown on apoptosis, proliferation, and migration, suggesting that KLC1's regulation of mitophagy is a key mechanism in NSCLP development. In summary, our study used TWAS approach to identify key genes associated to NSCLP. We found that the rs1288480G2A variant increased NSCLP risk by enhancing the binding of transcription factor ELK1 to the KLC1 promoter, which upregulated KLC1 expression. On the other hand, reduced KLC1 expression promoted mitophagy, which could lead to decreased cell proliferation and migration, increased apoptosis, and consequently a higher risk of NSCLP. These findings offer valuable insights into the genetic mechanisms behind NSCLP and suggest potential targets for prevention and treatment.